Dear Dr. Altuna Akalın and our esteemed participants, a warm welcome to Bioinfocongress VII. We are truly honored to have Dr. Altuna Akalın with us today, presenting on AI-assisted data analysis for biology. After the presentation, we will have a question and answer session with questions from both our team and our participants. We look forward to your contributions. Dr. Akalın, welcome once again.
Thank you.
If you are ready, you can start your presentation.
So I can do the full screen. Can you see it now?
Because yeah, we can see it right now. Okay. Everything is okay.
So I start. Thank you. Thank you for the invitation. I wanted to talk about recent developments for AI-assisted data analysis. This is a topic that our lab spends quite a bit of time. So I want to give you a brief background on our lab and projects, background on the drug development process. This is one of the main areas of AI-assisted data analysis topics for us. And then I'll talk about how AI can improve preclinical, clinical trials, target discovery, clinical decision-making, and general data analysis workflows. So I'll give a lot of examples. So to start with, who are we, what are we trying to do? So we in the lab are composed of scientists, software developers, and system admins. We are fully dry lab. When we want to do experiments, we either collaborate or outsource them to companies. We are interested in improving drug development and diagnostics using machine learning and omics technologies, but recently also other data modes. We also like to build reproducible, robust software so other people can use it in a reproducible manner. And we are located at Berlin Institute for Medical Systems Biology at Max Delbrück Center. This is one of the, I think, biggest institutes in Germany for molecular biology. So the projects in the lab started a long time ago, more than 10 years now, with simple omics analysis. So this was mostly bulk sequencing, mostly RNA sequencing or epigenomics related data sets. So at the time we had one data set per entity. So this would come from a biopsy or disease model. Then with the single cell technologies coming in, now we have one data set per cell in an entity. So you can imagine in a biopsy or blood sample, now you can look at not the bulk, but individual cells. And now we are also, quite a couple of years ago, we entered the era of spatial biology, where now we can look at each cell in its physical location in the entity. So this would be, given a pathology slide, you can, in the slides, in the correct location of each cell, you can characterize their molecular profile, such as RNA-seq or any other omic technology. And then you can observe this in place, so in relation to other cells in the neighborhood. And we want to use all this data to improve biomarker diagnostics and drug development, but we have to use AI for this purpose, because otherwise we cannot make sense of data, because it's huge data sets. But now we're also coming into contact with clinical text data, because if we are working on diseases or cancer specifically, we are interested in then connecting our findings with disease or clinical findings. So this comes in a clinical text format. On top of this, if we are doing spatial biology, now we have to deal with also pathology images, which is usually subsections of those images goes to spatial omics pipelines. And now we have to align them, so we have to also be proficient in image analysis. So all these things are combining, and we are able to use all these different data modes to make predictions or to make tools. So in cancer, we are interested in target ID. So what are good targets or target combinations for a given cancer? So you give us a cancer data set. We figure out which targets or which target combinations would work for them. Then we ask, can we repurpose or design treatments for those targets? And then we also ask, what are the good biomarkers for treatment response? For example, when you give a treatment to a patient, who would respond and who would not respond. So if you can predict this beforehand, then you can save time, money, effort, also sometimes patients' lives by sparing them from unnecessary treatments. The other thing we are interested in the lab is cardiovascular diseases. So we are looking for non-invasive markers that inform treatment. We want to optimize cost and timelines for diagnostics. Some of the cardiovascular diseases, such as subtypes of heart failure, for example, it's very hard to diagnose, takes sometimes years to diagnose. So we are looking into non-invasive diagnostics in the form of cell-free DNA or RNA that we obtain from urine or blood. So that's another focus area. But I'm mostly going to talk about cancer projects today. And our grand vision to achieve all these projects is that to build an AI assistant to help speed up the research. So we would like to ask questions like, what would happen if I knock out ISL1 in a neuroblastoma cell line or cell type with attached expression profile? Then I can answer these questions, by doing literature search, by doing deploying some of the scripts that we have developed or models that we have developed, and then I can give you a predicted answer. So in a cell with such molecular profile, ISL1 knockout is predicted to lead to cell death. So I can give you this answer already by, as I said, combining data from literature, from our models, from our algorithms. But the question is, can AI do this itself, or at least do part of it? So to be able to do this, then you need a database of molecular data points that comes from PDX, cell lines, tumors. You need literature that the model or AI assistant should be able to search the literature. Then you need to create data repositories and code repositories, so that's available for the AI to use. So that's of course, one of the main goals in the lab, but we are getting close to it, but we are not at this stage yet. But I think this is completely possible to build such an assistant. I will show you some examples towards the end. And now I want to give you a background on drug development pipeline and what happens during drug development and how AI can improve this. So it usually starts with target ID. So you need to find the target, which is a gene or protein. When engaged, it should stop or reverse the disease. So this is the first thing one should find. The other thing, after that, you find a good target, then you need to find an entity or molecular entity that can engage with the target. So it could be a small molecule, it could be a protein, it could be also CRISPR-Cas systems these days that can knock in genes, etc., to modulate the target so that change its function or function the disease. Then once you have a drug, then you go into clinical or preclinical development, which you test the candidates. So you usually have not only one drug at this stage, you have multiple candidates, and you test them on preclinical models. Are they effective? Do they reverse the disease in the model? Are they toxic? These kind of things one has to answer. And the clinical development, and now you have to test the drugs on patients to assess efficacy, safety, and for cancer and many other diseases, it's important to have a molecular biomarker that would make the patient eligible for a given drug. So for cancer, this is usually molecular alterations, mutations, etc. that drug targets, but sometimes it could be proxy biomarkers, which is not directly related to the drug, but it's been shown to differentiate the responders from non-responders. And after that, there is post-market, which means that after the drug is on the market, you can still do phase 4, it's called, trials to measure long-term efficacy, safety, and then at this stage, you can also try to find new indications. For example, if you're a cancer drug that's approved for lung cancer, now maybe you can try at this stage on other cancers or other indications, other subtypes of the same cancer, etc. These are done at that stage. So the challenges for this is basically high cost, long timelines, and high failure rate. So this funnel chart shows to get, you start with 24 candidates approximately, and then at each stage of the drug development, you sort of eliminate some of the candidates because they are not effective, they are not working, etc. But in the end, 24 candidates gives you approximately one successful candidate, and each phase of the drug development will cost millions of dollars. So approximately it could cost in total from 1 billion to 2 billion or more to develop a single drug. And if you are talking about cancer, 50% of the patients eligible for a new cancer drug won't benefit from the drug. So that's another issue, that even because of lack of understanding of how drugs actually work, we are not able to prescribe them to correct patients, and there's also 10 billion waste in treatment costs as well. So this is also something to improve. So what would a perfect AI system do? Of course, it would get the correct targets every time. Once you have a correct target, you can design, using this AI systems, perfect drugs that are efficacious, safe, can be absorbed, will be optimum. Then, since you have done perfect in the previous stages, you will have high success rate in clinical trials. You would exactly know who would benefit from those drugs. Of course, there's few problems with this. We don't understand the biology that well. We don't have the data for some of the AI models to train such things. And actually, even if we have drugs, we don't understand how they actually work. So instead of trying to figure out the perfect system, we should just use AI as a tool and to improve the timelines and costs, basically. And then we keep iterating, getting more data, training better models. This should be the sort of iterative process rather than right now hoping that we will find the perfect AI system to fix all our problems. So how AI can improve clinical and preclinical trials? So basically, you are focusing on these stages: preclinical, phase 1, phase 2. So if you select a more appropriate population and disease models as early as possible, you can then avoid costly failures. Basically, if you, as early as possible, if you can figure out which population will benefit from this drug, then you can design better trials. Also, you can even design or figure out better market share for a drug as well. As I said, the problem, this is one of the papers that came out some years ago, that they take 10 drugs that some of them were currently or at the time going under clinical trials. They knocked out their targets, or the targets that are reported by the companies or whoever developed those drugs. And they realized the drugs kept working. So efficacy of each drug was unaffected by the loss of its target. So these compounds kill the cells via off-target effects that are not studied, not even sometimes reported, or not known. So to be able to deal with such cases, as I said, we can integrate multimodal data as early as possible. So in this sort of set of problems we are dealing with, so we can integrate public patient data, we can integrate disease model data. So this is approximately about 1 billion data points. So we had the first shot at this in 2017. We developed a deep learning data integration model that can integrate lots of omics data, regardless of where it's coming from. So it could be patient or disease models, and then you would get these molecular patterns or latent factors from this deep learning model, and you can use those patterns to refine or discover subtypes. You can, when a new tumor comes, you can map the tumor to its neighbors to figure out if it's a if you have information about the neighbors, for example, therapy, response, etc. So you can interpolate from there. You can use the same patterns to model drug response, survival, and you can also interpret the patterns. So you can figure out which sub-networks or pathways that make up those patterns. And the intuitive explanation for these latent factors is that if we deploy the same algorithm on the pictures of faces, then the latent factors would be facial features. So it will be hair color, pose, smile angle, head tilt, eye color. These will be all captured in latent factors. But for biology or molecular biology, it's a bit more complicated. So for multiomics, the latent factors would be sometimes people call it composite biomarkers, fingerprints, but they would basically represent a set of genes with a common pattern or set of regions in the genome with a common pattern. And the pattern could be non-linear. So that's the advantage of using deep learning. So the idea would be here that then we can integrate labeled and unlabeled data across disease models and patients. So here on the left-hand side, you have tumor models. If you cluster them using some PCA or something like this, then you would get this, all the disease models will cluster together, same as primary tumors, so they will not be clustering perfectly based on subtype, etc. But if you have this integrated avatar which basically learns from labeled and unlabeled data at the same time, then you can figure out common patterns between disease models and primary tumors. And then you can sort of interpolate what you learned. For example, in this case, we have some response data for some of the models and disease primary tumors. And then we can then interpolate if a data set that has now label in terms of treatment response, then we can interpolate if it's going to respond or not based on its neighbors, for example, in the simplest explanation. So with such an experiment, actually, real-life experiment, so we have data from a lymphoma clinical trial. So cancer trials usually don't have large cohorts, especially if they're investigator-initiated. And to better understand response resistance, we can integrate the unlabeled data with small size labeled data from the trial. So in this case, we have 88 patients from the trial who has an outcome. So these patients have multiomics data plus outcome data. So did they respond or not? And we have from the same cancer, we have 1,300 plus patients without the treatment outcome, but multiomics data. So like in the fashion that I showed you before, you can integrate them and learn joint molecular patterns and then use those molecular patterns to predict who is going to respond or not. And if you do this exercise of learning from unlabeled data, your accuracy of predicting who's going to respond or not increases. So this is actually the test. So if you just use the clinical trial data, so there's still good separation. But if you use the additional 1,300 data that are unlabeled, so the separation improves. So accuracy goes from 80% to 92%. So this is quite significant in terms of who is going to respond or not. So any improvement is welcome, and such improvement is even more welcome because the accuracy increased more than 10% in this case. So this is a work by talented scientist in the lab, Bora Uyar. He, of course, all the you can use it. We gave a course recently about this. There's a Python package. So this workflow basically helps you do data cleanup, feature selection. You can do hyperparameter optimization. You can have different models, different deep learning models, also off-the-shelf non-deep learning models. You can benchmark against them at the same time. You have different tasks, so you can do survival, clustering, classification, etc. You can also choose to fuse the data early or late. So all these things are embedded in the package. So you can basically start using it today. You can just go to GitHub. There's a preprint on it. We are also submitted a more recent version. So you can play with it. There's already good tutorials. And if you want to do multiomics integration, it's a good I think, good toolkit, very reproducible and usable, I would say. So, of course, single cell and spatial omics, as I mentioned in the beginning, becomes more and more important. For this tasks, we have a nice R package that is developed by Arthur Manukyan, who is also a very talented scientist. In this case, we wanted to develop something that all spatial technologies can use. So regardless of if you're looking at spots, cells, or molecules, or tiles, you can integrate data, because in the spatial omics field, in the beginning, it was, let's look at one slice of a pathology sample at a time, but now we are even trying to build 3D models of the tumors, or one slice comes from RNA-seq, the other slice comes from a different technology, then you still want to integrate them. So for this, all this kind of integration work and downstream analysis, you need something flexible, fast, etc. So what we have, or what Arthur developed, is this unified workflow across spatial resolutions. So you can have different technologies, resolutions, you can still combine them and analyze them together. You have this automated data alignment strategy. It's quite fast because we use C++ in the background. And it's most importantly, it's interoperable with other technologies. So if you want to use Seurat downstream or SquidPy, you can convert objects between different things. And even more importantly, we use highly efficient data structures like ZAR and HDF to make things a bit faster, because all these data sets, even the imaging data sets, are huge. They need some efficient on-disk storage and query data structures, which is, I think, HDF or ZAR these days. So it's all integrated. And of course, you can do downstream analysis, you can do multi-omic integration, you can visualize it, etc. Again, this is a fully fledged tool. We use it in the lab, other people use it. There are lots of tutorials on it already in the website. You can start using it immediately. And this is a small video on what it can do. So this is a molecular resolution analysis for the SARS-CoV-2 transcripts. You can do downstream analysis like clustering. Everything is integrated. You can do this alignment of the pathology slides. It can do automated alignment of different image types. You can do interactive visualization, so color code the slide based on, in this case, cell types, etc. Yes, so the next question is how we can improve the target discovery. So target discovery starts very early on, before you start to generate hits for those targets. They are called hits, so initial molecules that engage with the target. So in this case, we want to know which targets are valid in primary samples and models and optimize experiments. So meaning that, first of all, you need to select good disease models that represent the disease. So if you're talking about cancer, there are lots of disease models. Some are better than others. All of them are questionable. But again, you have to select the best one that's available. And on top of that, you have to select the target that should modulate the disease, not only in the model, but also in the primary sample. So you need to integrate, at least have an understanding of not only of the disease model, but is this target valid in the primary disease or primary tumors or not? You have to ask these questions. The target has to be specific, so should be specific to the disease. Should have an effect on the disease when perturbed. And what we have come up with is a use of customized prior information to simulate an in silico experiment. So in this case, we take a prior gene network that's curated from the literature. We customize it based on the data from primary tumors. In this case, this is single cell data from a tumor or tumor type. And using the customized network, now we can do sort of in silico perturbation experiments where we knock out or in silico knock out targets and see what would happen. So then we can select targets. So if we can do in silico prioritization, then we basically save costs and timelines, because then we have to go to next step, which is the validation phase for those targets. And the fewer targets we want to validate, the less time it takes and the less money we spend. So to test the validity, we did this double knockout experiment using CRISPR. So we have this, in this case, 3 neuroblastoma cell lines, which was the target disease in this case. And then basically we did this CRISPR experiment to see if the targets we selected and some other targets that given this control would have an effect on the disease or not. So this is the resulting model. So this is the measured effect of the target being essential for the disease on the y-axis and the predicted effect. So there is a high correlation between our targets or predictions and the targets. And then that means, of course, then we don't have to do thousands of experiments. Maybe we can do, from our predictions, we will take the top 10 and do experiments on them only. So this saves us time and money. So of course, how far we are away from making a drug. So if you go to an investor or anyone who wants to develop drugs, they want a data package. So the data package should have at least mouse studies. So after target discovery, then you need to devise a drug, which we're already in the process of it, but then you need to test them on the mouse models, for example, which are far from, I mean, it will take maybe 2-3 more years. So we are far away from a visible impact. But in the meantime, we also ask ourselves, what can we do that is patient or clinician facing? And also we can, all the tools are developed, some of them, of course, open source, you can already use it, but also we can make a platform and the tools around it so more people can use these tools. That's the idea. So 2 ways of making things more accessible and usable in the real world beyond the drug development. So one thing, as I said, is the AI improvement in clinical decisions. So this focuses on the last part. So as I said, 50% of the patients who are eligible for a drug won't benefit from the drug. This wastes time, money, lives. And for the cancer specifically, there are new generation therapies called targeted therapies. They target an alteration, a specific alteration in the genome or in the protein part of the molecular profile. So if you have this target or a biomarker that's related with the target, you can get this targeted therapy. So sometimes, I mean, in the past years, they were only given to people who have gone into first round of chemotherapy or surgery, etc. But now, at least in the US, this targeted therapy is coming in the first line. So instead of getting chemotherapy, you can get the targeted therapy first if you're the target for some diseases. I don't know, in Turkey or Europe, it's not that common. They usually want you to go through the guideline first, which is usually chemotherapy, surgery, radiotherapy, or something. Then, as an add-on, they would do targeted therapy, or they would do in the second round or something. But in any case, if you don't have a target, if there is no target for any drug in the market, then you get conventional therapy. So these are the sort of the targets or biomarkers that are associated with the target. On the y-axis, there is the response rate to those targets and the prevalence of the disease. There are different diseases color-coded, but I'm just showing you to tell you that there are many targets, there are many more coming, and for each target, there are multiple drugs that are available on the market. There are 3-4 times of that in clinical trials, so that's a complicated landscape. Sometimes you have 2 targets available in a patient. Which one you would give? It's a question. And on top of this, each cancer is a guideline, especially in US, in Germany, for sure in Turkey too. There is a guideline that clinicians should follow. And this guideline changes frequently sometimes. Basically, it's a complex decision tree based on the stage, clinical markers, or information. You decide what therapy patients should get. It's also difficult to traverse. So if you're going to apply the targeted therapy, it's hard to navigate for patients and doctors. The guidelines has to be kept in mind. Also, which targeted therapies are available for a patient? It changes. New biomarkers come. You can also sometimes give off-label drugs to patients, so you need to do some research to figure this out. Also, as I said, there are many clinical trials for targeted therapy. Which ones you would fit? Which ones are available in your area? That's a question. And of course, what's the latest research on a given cancer? So usually doctors don't have time to read a lot of research papers, but that's why AI comes in handy, that can search, given the disease profile, it can search what's relevant in the literature for that disease profile. So keeping in this mind, we generated a customized language model that has the guidelines, markers, biomarkers, it says clinical trials and PubMed all embedded, so it can answer questions like match the patient profile to a therapy based on the guidelines. If the patient profile already has molecular biomarkers or molecular profile, it can find which drugs are eligible on and off label. It can find clinical trials that match for a patient based on the biomarkers, and then it can also search the literature at the same time. So this is already available. It's a search engine for cancer patients and clinicians, so you can enter your patient profile and you would get some response. So it's a live website you can go in, but I have a short demo where we do the first query. So this is lung cancer with BRAF V600 mutation. So basically when you search, you find the guideline first. So based on the guidelines, MEK inhibitor, which is correct, is an option. So median survival and risk based on the guidelines. You have the approved drugs that are specific to a cancer, to the lung cancer. Also, you can also look at other cancers that are approved for the same biomarker. We had the, maybe I can go back a bit to show you the. So these are the all eligible therapies that are running at the same time for this kind of patient. And there is also the short literature on treatment. So what the literature says about this cancer, some information regarding that. But after, so next question would be something without the biomarkers, because most of the actual cancer patients won't get sequenced and they will not have a biomarker that can be matched with a biomarker database. But in those cases as well, the platform works. So this is a lymphoma patient. There's only information about age, stage, etc. But again, you can search the guidelines based on that. So in this case, for this kind of patient, R-CHOP treatment is suitable, etc. So all this information is there. You can also click on the links in the guidelines to see exactly what the guideline says for that specific sentence. You can find, again, approved drugs if it matches. And again, you can find clinical trials and some information from the literature. Yep. So the performance, so this is ongoing. So we tested on 10 lymphoma cases, 11 lymphoma cases, 10 out of 11 agreed with the tumor board suggestion. We are actually doing a bigger or larger validation study right now, more than I think 200-300 patients, and in collaboration with clinicians to see how well this kind of system matches the expectations of clinicians. There's a preprint on the clinical trial matching. I think on the guideline-based matching, as I said, we are doing more benchmarks at the moment. But there's a more generalized version of this. It's called second opinion. So after we built Onconaut, we said we can also use this for all medicine, not only the cancer part. And this is again a PubMed-driven search tool. It's already available, secondopinion.io. So also available in Turkish and other languages, but you can basically, what the main idea is that you have some medical conditions or symptoms, you can put them in, you can put your blood test results or whatever also as text, or you can upload them. So we don't keep anything uploaded, everything is deleted after we upload them, and so we don't keep any personal data, but also you can type your results as text here as well. But the main idea here is that once you go to a doctor, especially in US, it's very common to get second, even third opinion about your case. So this gives a sort of automated second opinion. Also, you can use it as a first opinion if you don't have any data at hand. You can just write your symptoms. But in this case, we basically wrote. So let's read this case a bit. So this patient has exercise-induced angina. So this is chest pain. Had never such complaints. She was hospitalized. Suspicion of heart attack. His heart attack is clear, but hospital stay reveals left bundle branch block. So this is a sort of a electrostatic, I think, mismatch in heart that causes the flutter. So angiography and MRI revealed congenital narrowing of the right artery. There's no myocarditis. So these are all patient information that comes from the patient journey. And then once we put this in, so she also had a COVID infection. So what comes out of this is that there are a lot of new papers coming out. COVID infection basically induces some heart diseases. In some cases, there are also clinical trials to treat those cases. And actually, in this case, we found a paper through here that suggested a medication that would work for such patients. And actually, for that patient, this kind of medication worked because also it's a new information. So such cases, the rare cases that are hard for the doctors to figure out because they don't have time to keep up with the literature, is very beneficial. We also actually use it whenever we have a medical question or we have some lab results that we want to confirm, because if you get a lab result, you just get a table or something. You cannot really decipher it all the time. But with such tools, you can understand what it's saying. You can understand what the implications could be, and you can see if you should be worried or not. So other applications of this is that, for example, reducing unnecessary ER visits. Many of the ER visits are not necessary, so it clogs up the system. So we are benchmarking different things on this system, or such systems actually. There's a preprint on it. We are benchmarking ER visits. So can it do accurate triage? So who should go to ER or not? We are testing what kind of, can it predict the specialty? So in some health systems, you can directly go to a specialist. In Europe, or many places in Europe, you should first go to a general practitioner. You cannot just go to a specialist, but in Turkey, in some of the Eastern Europe, you can just pick a specialty. But then it's on you to pick the right specialty. But these kind of systems are accurate at predicting which specialty you should go based on your symptoms. The other thing is diagnostic accuracy and ER visits. The other thing we noticed is that since it uses PubMed, and PubMed also has lots of veterinary articles, you can also check the pet symptoms or animal symptoms. So if you have pets, they have some symptoms, you can write it in, and actually we diagnosed not our pet, but our relative's pet correctly using this kind of system based on her symptoms. Yes, so the other thing we can do in the meantime, so we can build patient-clinician facing features, give access to them, but also we can build a system or platform that has the tools that we developed in them to do research. So the interface question, what would be the interface of the platform? As I said in the beginning, you ask a question, you get an answer back. So it's a chat-based platform would be natural. So the vision would be exactly like in Star Trek. If you watch that show, you ask a question, a scientific question to the computer, and the computer responds in doing some analysis, and then gets you the answer. So that could be the idea. So first proof of concept, there is an R package we built, or two R packages that is basically is able to understand your question, see your data, and do something on it. So in this case, example is that you have a gene expression table called leukemiax.txt. The table has genes, etc. And the task is to filter 30 most variable genes and make a heat map. So the system we have sort of reads your data. So it's a RStudio add-in. So it is integrated into RStudio. So it can see your data, read it in, execute it, and give you this plot on the right-hand side. So stuff in it is that it can do self-correct. So if it gets an error, it can try to fix it. You need to give an LLM API. So it could be GPT or any other API that you can get access to. Some of them are free. It augments your prompt. So basically, it can use your data itself to write a better prompt so that the output is more precise. And of course, this is not perfect. So when we started this, it was much worse than this, but the code we got was sometimes completely useless, sometimes perfect, but somewhere in the middle. I think we are going, with each new model, we are going towards the green point. So at the time, GPT-4 and O was somewhere here. I mean, Claude, etc., there will be a bit better, but not, especially for our stuff, they are not perfect. And I'll show you why. So if you do prompt engineering, so write detailed prompts. If you have some background knowledge, you have to write it in, and you can get better results. So we published this this package and our experiences with the large language models for data analysis in a paper. And also the Mergen and Mergen Studio is all publicly available. You can try it out. It integrates with RStudio. You have a chatbot on the side. So you can also domain train this. So you can do either RAG or something fine-tuned. Then it will get better, of course, for if you want to analyze genomics data. So hallucination is a problem, and still a problem. So it takes time to figure this out. If you domain train it, you get less hallucinations, but I think if you're not an expert, maybe it's if you get some weird results, it's better to ask an expert at some point. But usually for simple stuff, not simple, but routine stuff, it works well in our hands. So this is the first version. Some examples I want to show you here. Get a heat map. This is the interface, the R interface. This is the same thing. So leukemia expression profile, you have genes, get me the 30 topmost variable and make a heat map. I'm going to fast forward. So you get the explanation and the code. You can execute it. You can auto-execute it. It's up to you. In this case, we execute the response and we get the heat map that we are looking for. Let's move forward. The other thing I wanted to show you is the PCA. So the next one would be PCA. This is a very common task. You get the data, you want to do PCA only to see how samples or features relate to it. So this is a cancer sample. In this case, you have two data sets. One has the expressions, the other one has the cancer subtypes. So it has to correctly merge them or use them. It's sometimes difficult for this kind of things to do it properly. And in this case, yes, you get your subtype-based PCA plot. So these are different cancer subtypes. It's correct. The other thing, you can do more genomic tasks, such as plotting ChIP-seq peaks across a chromosome. In this case, you have a bed file. You want to visualize them on the chromosome 20. And again, you So for these things, we are using our own API. I don't know what happens if you use GPT or something. You can try it out, but I cannot say it will work out of the box. So in this case, you get the code, you get an explanation, and then in the end, you get also the plot. So you can do different things as well, but this is something you can try out. In the meantime, so we also compared, as I said, we have our own API to do this kind of tasks, because we know that if you use something out of the box, they are not specifically trained for our tasks, and they will not perform. And the performance will depend on how easy is the task. So if it's easy, they will be okay. 1, 2 tasks. So basically, read the file, give me the most common variables or something, or most variable rows. These are easy tasks. But the complexity increases when you use multiple files, when you ask the system to do data filtering, data merging, and then on top of this visualization and applying some machine learning model, then you'll start to get errors, because it either doesn't see your data properly, it's not, the prompt is not, because of that, the prompt is not engineered properly. So to deal with these kind of things, we developed our own API. So these are on the y-axis is the fraction of executable tasks with correct code, which means what percentage of the tasks that we gave has a correct code that executes. So as you can see, this is 3.5. So 4 is probably a bit better, but not as good as our blue. You can see it's higher, especially on the highly complex tasks. So this API is available. We did some training on it a couple of weeks ago. We will give large-scale access at some point, hopefully in a couple of months. You can request access if you go to this URL. And also we are building a more standalone app that runs on your own computer without RStudio. The benefit of this is that now we have more capabilities. So instead of now we are going to do differential expression. So you have a TGF-beta RNA-seq expression. The 3 columns are controls. The rest are the TGF-beta knockout. So it says, give me top differential expressed genes. So do differential expressed analysis, and then give me the top 30 differential expressed genes in a heat map. So I'm going to fast forward. It does these things. It gives you the heat map. But on top of this, right now, we want to add more capabilities, because after you do all this data analysis, now you want to find more data sets. You want to do some literature search. For example, you can start a new tab. Snail 1, you can start a new tab, and for example, there's an interesting gene in your heat map, you can search for it. So this is Snail 1. What does it do? What are the known functions? So basically, it searches the PubMed, gives you references, what this gene does. And on top of this, now next thing, once you have developed an idea, you want to do addition analysis. So you want to find data sets, then you have to search for data sets. Then another functionality, if you say, find me DNA binding chip-seq data for Snail on Geo, so it starts to try to find similar data sets on Geo, the ones that matches your criteria. So it finds some, in this case, it couldn't find exactly one, but related ones, I think, and gives you the responses or results. So there's some more work to be done. So you can also imagine that you can even download those datasets and start working on them. So that's the next step for us to be more integrated. So the other question would be, if you are doing a drug development or some exercise on what could target TGF-beta, it can also do some literature search. Yeah, so targets, sorry, it's not about drug targets, it's about the targets of TGF-beta itself. Then again, once you find something interesting, you want to go deeper in the literature to figure out what it is. But also you can incorporate other databases. via agents to this platform where it searches for PubChem, for example. Then you can ask, are there any known molecules that engage with TGF-beta or anything on the pathway? Then it will start searching available databases to figure out what would be the drugs or drug candidates that already shown to engage with this pathway or the protein itself. So that's another thing we want to achieve, that basically all these searches and data integration and stuff can be assisted by the AI. Normally it takes a lot of time, these things, but now if you can integrate everything, you can get answers quickly. And that's the point I made in the beginning. Drug development takes time, expensive, but some of this thing can be outsourced to AI agents. So normally our workflow is that we have a PI, a PhD master student, we discuss project. Once we decide on something, the student has to go through public data, methods, original data. That takes 1 to 3 years. If we have a postdoc who is experienced, maybe he already knows methods, he is a sort of intuition about data, already learned, it takes less time. Sometimes we have teams, which works quite well. In our case, then we have the tasks are distributed to team members, and then the execution time or iteration time is shorter. And what we face regularly is that I'm going to give an example. So, for example, there is a project to integrate prior knowledge to deep learning applications for omics data analysis. The student has to figure out which gene networks to use as prior knowledge, which data sets to test, which deep learning architecture to use. There are many of them. It has to be all experimented. What other methods are available? So you have to benchmark this against other methods that are simpler, maybe still might work better than the deep learning, which sometimes or maybe often the case. So student has to go through this. If something doesn't work out, then we have to figure out, is it my fault or is it the fault of the data quality or selection, or it's a bad idea from the beginning? So this whole iteration takes a lot of time, especially if the student is unexperienced. It takes even more time. Also, there's a mental load to this kind of failures, which is normal in research. But if the student is new or unexperienced, it can think that nothing works out, could go into depression, etc. But I think if we want to iterate faster, then we need to use this AI help. For example, initial feasibility and dataset selection can be done with the help of the agent. Also, the students or everyone else have now AI agents under their can use them, and then they can cycle through the ideas faster, at least a portion of it. It cannot replace everything that we do, but at least you can do the literature search faster, you can initiate, you can test your ideas against literature faster, you can do all these things a bit faster and integrated manner than just going through it on your own. And I think that's basically that's the vision that we have, that we will use these tools, we will build those tools as an assistant for us to iterate through ideas faster. As I said, this can be a useful assistant for anyone in the life sciences or clinics research, but of course, I don't think it can replace humans in any way, but I think if people use it, they will be more efficient, and people who don't use it, they will be less efficient, and that's the problem. People who use it will be selected for, and people who don't use it will be selected against. So these are the tools I mentioned. I mean, if you can take a picture or whatever, they're all available. So yeah, so we are looking for academic and industry partners to deploy second opinion. So we have collaborations in Poland to deploy this in clinics to see if real life accuracy and how people view it. We want to do this in Turkey as well. So far we don't have any collaborators. We have open source projects. Most of the projects are open source. It's in this GitHub. We of course always look for academic industry partners for grants, code development, etc. So this is the lab. So most of them contribute to many of these projects. If you have questions, I'm happy to answer them. I don't know if I'm over time or not. Probably not. Thank you so much.
Thank you very much, Dr. Akalın, for this informative presentation. We have time for actually question and answer session, at least 15 minutes or something. So we can ask questions if you want.
Should I? Where do I see the questions?
Yeah, I will ask you.
Okay, you ask them. Okay, good.
Yeah, considering the black box problem in AI during diagnostic and therapeutic processes, where decision-making processes are not fully understood, how clinicians trust in these technologies be improved? What transparent AI models or explainable AI approaches would you propose to address this issue?
Yes, I mean, trust comes from benchmarking those models, especially in clinics. I understand there is a big push against AI, but again, I also lived firsthand the inability of doctors to make the right decision because they don't read, they don't have access to information, they don't have time to read about cases that are a bit trickier than normal. So I think they definitely should look into using this as a tool, not as a replacement. So I wouldn't also trust just AI-based diagnostics, but again, doctors have many blind sides. They don't have time, especially countries like Turkey. If you go to public hospitals, there's 5 minutes or less for a doctor to see you or diagnose you. But some of these things can be alleviated by using AI-based assistants to search through symptoms, search through potential cases, and the doctors use this information to guide the decision. It's not the final decision is still by the doctor, but it's maybe more accurate and quicker if AI is used. So the trust in AI will come by benchmarking for the drug development. It's important to know this important features, etc. There's a lot of, we also do it in our case. We try to explain features that are influential, etc. Those things are very important for drug development, but I think for the clinics, I think it should come with benchmarking. So what is the model's accuracy on a given disease that is displayed to the doctor along with all the information the model finds, especially on rare cases. I think that's the way. And the doctor still makes a decision, but then he or she has a second pair of eyes that looks at the same case with from a different angle with a lot more data than the doctor has time to sift through. I think that's the way I want this to work out. I don't know how it will work out, but it's my vision.
Thank you for your answer. The next question is coming from our YouTube participants. Do deep learning methods truly outperform traditional machine learning methods in multi-mix data integration?
Sometimes.
In the comparison made by Flexinesis, in which scenarios do traditional methods perform better?
Yes. So Flexinesis paper has lots of benchmarks when this performs better or not. So you can read it from the preprint. In our experience, not always the case. That's why we put this off-the-shelf benchmark models to see if they truly outperform. In the cases, for example, this augmentation of unlabeled data is very difficult with the traditional methods. I think that's one case that it helps. The other cases, we are also on the fence. We are not full deep learning because we also observe that sometimes just PCA works, and that's why people should try different things. That's why we built, or Bora built, Flexinesis to account for these things, so that whatever method works on your data better, you use that, and everything is included in Flexinesis. That's the answer, I think, to that question. So deep learning is not always better, especially if you don't have so much data, and especially the trends are so obvious that the linear methods can easily figure them out. Yeah.
Thank you for your answer. The next question from our team. What advantages do AI-based variant calling tools, example deep variants, offer over traditional methods in the analysis of VGS and VGS data? And what strategies would you recommend to minimize false positive rates with these tools?
We then never did deep variant, so I cannot comment on how good it is or not. So this question I pass on. Yeah, pass.
Okay, so the next question is from YouTube participants. In CRISPR knockout studies, could the cell's abilities to adapt the gene loss lead to misinterpretation of results?
So could the cell's ability to adapt? I mean, if you do it in a replicated manner, it's less likely, which we do. And I think I'm more worried about not cells' ability to compensate, but cells' ability, the off targets that we are not fully sure about the CRISPR experiments. But that's why we did two rounds, multiple replicates, but also multiple technologies. So we did not only knockout, we also did this CRISPR-I, which I didn't mention, which shows the same results. So there is a potential, but by doing this multiple times in different technologies, we are more sure.
Thank you for your response. The next question is again from our YouTube participants. How can transfer learning and pre-training models open new research areas in multiomics data analysis? How does Flexinesis facilitate the adaptation of these models to clinical data?
I think one thing we should do, which is not in Flexinesis, is this pre-trained models, for example, these foundation models or transformers. We don't have transformer architecture in Flexinesis yet, and that would be, I think, helpful if you can fine-tune a pre-trained model based on your data set. That would be interesting to do. I think that's the way to test those things. There are people trying to do those things. Right now, it's a bit tricky because there's no unified infrastructure. Every model has its own quirks. So if you want to fine-tune a model and it's not well documented, etc., it's also a time sink for people. But I think that's what we and other people are doing. So use pre-trained models to fine-tune based on your data and use that. So that's the right way to do it. What would facilitate in clinical data? I don't know. It's the same thing as before. Probably you will have. I mean, the idea is that you will find the biomarkers, etc. It's always the same. The things you are trying to look at. Will it be better than what we are doing right now? I don't know. That's why we have to test it.
I see. Thank you for your response. The next question is, what are the bottlenecks in predicting drug response using genomic features obtained through comprehensive genomic profiling methods for use during clinical and preclinical drug development?
I think one bottleneck we observe is that preclinical and clinical data that's generated through clinical trials and preclinical experiments is usually shallow. So they use not whole genome sequencing even, they use targeted sequencing. We observed and other people observed that transcriptome is much more important in predicting drug response, which doesn't usually come in most, maybe I will say most of the clinical trials. And that's the bottleneck. So they have limited amount of molecular data, and I don't think that is super predictive, especially when we think that the drugs are usually multi-target. We don't fully understand. Maybe there are many studies that, especially kinase inhibitors, they usually bind to multiple places. Maybe they bind to their target more strongly, but they bind to a bunch of other things that you don't account for. And that's why I think other data modalities are important, but they are expensive. They are not usually part of the clinical trials. maybe the new companies and new genomic-oriented companies are generating data like Roche or Genentech and stuff like that, but it's not common to have deep data, and that's, I think, the major bottleneck to do these things.
Thank you very much for your answer. The next question is, how can the rapid integration of updates, particularly those related to FDA approvals or new clinical studies, be ensured during the updating of AI algorithms? What potential obstacles might arise in the process?
Technically, there's no potential obstacle. You just have an AI agent that parses FDA website regularly and updates the database. If there's a regulatory obstacle, I don't know. There shouldn't be, because it's just a database. But technically, this is quite easy to do, because once FDA approves something, it's immediately announced. The only problem is that whatever FDA approves doesn't immediately come to Europe or Turkey. So that's the bottleneck, I guess. So even if something is approved in the US, doesn't mean that you have access to it in other parts of the world.
Mm-hmm. I see. Thank you for your answer. Another question is, could spatial targeting technologies such as nanocomposites, polymers, and fibers be used for cancer treatment?
Nanocomposite polymers. I never heard of this. Maybe. I mean, whatever. I cannot. Yeah. So I pass on this question as well, because I don't know what those new technologies are. Spatial targeting. So I pass.
Okay. Thank you. The next question is, although many preclinical models do not reflect the complexity of the actual tumor microenvironment, how can large-scale data analysis of drug responses be extended to real human samples?
This is very hard to do. I mean, especially for the drugs that target microenvironment, it's impossible. We have some success with some drugs that are predictive, but not everything is predictive. This is maybe due to the complexity of the tissue is not represented in the tumor models. People are trying to tackle this with organoids these days. So they're trying to build complex organoids that have microenvironment and stuff like that. That I think that might solve this issue, but again, there will be always some uncertainty when you have a model that should represent the reality, because it's just a model. A simplification of reality is always brings uncertainties when you test these things in clinics. But I think technically organoids probably is the way to go. Then cell lines. But again, cell lines are so ubiquitous, and it's the first thing people usually try. Among the cell lines, one should try to pick the one that represents the one best, even though the tumor best, even though there's no perfect cell line, because they lack the tumor microenvironment. They are sometimes quite different from the actual cancer. But again, between them, there is a hierarchy. So some of them are quite different, some of them are a bit closer. So pick the one that's closer. That's what we do.
Thank you for your answer. The next question is, while virtual reality-based simulation learning offers significant benefits in health education, what are the challenges in its full integration?
I don't know any virtual reality-based simulation of learning. Health education, meaning that I guess doctors are using this to do surgeries and stuff. I have no idea. So I passed this question because I don't understand it.
Okay, okay, thank you. The next question is, how reliable is the accuracy of large language models in addressing a domain-specific question, and how can they be improved?
So one thing is that I don't think they will be ever super accurate or specific. They can come close to being very good, I think, especially in some problems. It's very good, like simple differential expression. I will use it. If the task is complex, and you get errors, so you cannot execute it. Of course, there's a problem, then you know something is wrong. But the problem is when it executes and it gets the result, and the task is complex, then you don't know what you're getting. So then I think that has to be augmented by real humans. So in our platform, for those things, we want to put an ask a real human thing, where people can ask questions when they are not sure. So I'm not super optimistic on this being very accurate in all problems. That's why I think there should be an option to augment this with human or expert humans. That's what I can think of. Although I use it a lot, I can't say it will be as smart or as proficient as a real human who has looked at this type of data for decades. That's my vision on this. So there has to be some augmentation for complex cases. Maybe over time, when you have enough of complex cases that the system has seen and learned from, maybe. But yeah, there will be cases it always will make mistakes, I think.
I see. Thank you for your answer. The last question is from YouTube participants. How can early career biologists and bioinformaticians best prepare for the increasing role of AI in life sciences? What skills do you recommend they develop?
What skills? I mean, they should still learn the basics, like how to analyze data, etc. Try to use AI in the projects in terms of, I mean, the expertise should still be, so you should be able to determine if AI is accurate or not. So to be able to do this, you need some expertise. We are using this because it saves us time, and we can see that it's correct or not when we look at what it's giving us. So if you don't have this expertise, you will be relying on the answers of the AI solely, then of course that's a problem. So I think you should use it to train yourself, still do all the exercise, etc. But most importantly, for everyone, I can recommend to do real projects. Coursework is not enough. You need to do real projects in internships or elsewhere. The real projects, meaning that there is some possibility to publish something, a report, internal report, a paper. The more of those things you do, the better you're prepared for real life. And I can assure you, doing coursework is not enough to prepare for anything in real life. The most important thing is to do projects. And if you want to do bioinformatics, you need to do projects that you can then verify to an employer. So it's either on GitHub, it's either published. If it's on GitHub, make sure it's well documented. People can understand what you did. If it's published, make sure you get the credit for what you did. This is the most important thing. Do projects. Everything else is not important, I think. Grades are not important for me. Only projects are important.
Thank you for your answer. And it was our last question. Dear Dr. Akalın, thank you very much for taking the time to share your knowledge and for your informative presentation on behalf of myself, our team, and all our participants.
Yeah, thank you so much. I hope you learned something, and as I said, do projects. This is the most important thing.
Okay, we will, of course. Okay. We wish you all the best for your health and success in your studies. Thank you very much.
Thank you so much. Bye-bye.
Bye-bye.